Comfy. Good afternoon, everybody. Welcome to a Wednesday afternoon of Organic Chemistry, Chem 170, with your host, me, Dr. White. Hi, everybody. Excuse me. All right, important things. Tomorrow, I will be giving test number 4. And hold on, let me check something real quick with you here. I'm logging into D2L. All right, everybody, do you see D2L on your screen now? Thank you. And if we go to assignments area, you can see test number 4 plus some other files that you've seen before, and you can download it tomorrow right after lab. Remember I said, hopefully you got the email I sent out this morning, and I mentioned it yesterday in class, that today we'll do all lecture, and tomorrow we'll just do a lab, and right after the lab, I will send out the password for test number 4, which means that should be about 2 o'clock tomorrow. I'll be sending it out instead of 4 p.m., but you'll still have until, I believe, let's take a look, 11 a.m. Friday to take it, upload it to D2L. And let me log out of this, so I no longer need to, first of all, Oh. Anyways, that will be tomorrow. All right, let me give you a heads up what's happening. Today, I'm going to be going through the carbohydrate problem set. I did some work yesterday in carbohydrates review, or actually do some stuff. I'll finish up that today. That will help you for test 4. And then afterward, we'll finish up the last chapter. Amino peptides and amino acids, peptides, and proteins. After that, I've got a special gift for you today. I will be taking you on a tour of a chemical plant that I worked at. And I'll talk about that. And then afterward, we may get out a little early, because one of the things I haven't told you is I cheated this semester. How did I cheat? Well, normally, face to face. Hold on, I've got a frog or something in my throat. Let me get rid of it. There we go. I think I got it. Maybe not. But anyways, I'll take you to that plant, and we're done. And the way I cheated was, when we go face to face, when I give a test, that takes time out of when we meet together, an hour for each test. Well, the way I've done it here is we don't take an hour out for each test. I let you take it after class. So that's why sometimes we get a little ahead of ourselves or finish early. And that sometimes happens. All right, let's go right to a couple other things. Let me remind you, don't forget, if you haven't uploaded and submitted your labs, if you look at D2L, this morning I uploaded the lab that you handed in on Monday, the scores for those who did. If you're missing lab scores, and you don't have a lab, contact me. You have until Saturday. That's my lab amnesty. Hand something in, otherwise you get a 0. And labs are 11 points, and it adds up if you're missing a lot of labs. So this is your chance. Also, uh, don't forget, I have my office hour tonight. If you need help, stop by. I'm there to help you. And also, next week, the extra credit project, I believe, is due. Let me check something real quick. A calendar. And next Monday is the deadline, the 26th, for the extra credit project. The extra credit project is optional, but it's 10 points. It's like getting 10 points on any test extra. And it's a good thing. And remember, my public service to you is do the extra credit before you upload it to D2L. You can email it to me. I'll check and I'll say, no, this is wrong or this is wrong. And you can correct it so you get your 10 points. And it's not that hard to do, but if you want your 10 points, you have to do it. And coming attractions next week, next Monday, I will be going through test number 4, not practice, answers for the test, but I'll cut it out of the video. That's the way I operate. I will also, all next week, except when we have our lab, we will be doing a review for the whole semester. And that helps you do good on the final. And on Monday, I'll talk more about the final, but over the weekend or by Friday, Saturday morning, I'll have posted the points breakdown, and it'll be both by general knowledge, nomenclature, and reactions. And it'll also be material from test 1, 2, 3, and 4, and a little bit of the new stuff, which I'll talk about today. And that will be the final. I'll talk more about it on Monday. Next Thursday, since I'm giving the final, we'll have no class. What I'll do is I'll show up. If you want to stop by and say hello, you don't have to. But at 1 o'clock next Thursday, I will send out the password for the final. You'll have until Friday morning. Let's make it 10 a.m. or 11. I'm not sure which I'll do to get the final. Now, normally the final is 2 hours. Yes, it's comprehensive. Most students, when I give it face to face, are done in about an hour and 15 minutes, an hour and 20 minutes. And yes, it's a final, but students do really good on the final. Why? Because I do my review. All right, any questions before I move forward? All right, let's go through the carbohydrates and lipids problem set. All right, does everybody see that on the screen right now? Yes, thank you, Joanna. All right, now, what's a carbohydrate? Dr. White got lazy. C knows. A carbohydrate is a polyhydroxyketone or a polyhydroxyaldehyde or a compound upon hydrolysis becomes a polyhydroxyaldehyde or polyhydroxyketone. Number 2, in each of the following items, is each of the following items chiral or achiral? Remember, chiral, it has a non-superimposable mirror image, like your hands. My thumbs line up, the rest don't. If my middle finger, my thumbs and other fingers don't line up, but it's a mirror image, chiral. Now, if you have a achiral, I'm trying to, I used this the other day. And if you have two things that are mirror images, let's see, you can see it mirror, and they're superimposable. Notice metal, metal, black, black, center, black. These are mirror images superimposable. They are achiral. Now, important tip. If you ever see a name right or left, you immediately know it's a chiral molecule or a chiral object. So let's go back to the problem set. And as soon as I say your left foot, chiral. Now, mitten with no leather palm and also no writing on it, but no leather palm, it's a chiral. And look out, sports fans, it's time for me to see how hard I can make you laugh. And let's do a mitten, no palm. Like I said, I'll never go to art school. Here's the mitten, no fingers for it. And the mirror image would be this. And if you rotate this way, it's superimposable. Therefore, this is achiral without handedness. And if you think about it, if you have mittens without palms, you can wear them on either hand. And that's a good test too of chirality. Tube sock, same thing. If it's got no toes, I should have put also no toes for writing or design. It's achiral. Sewing needle, I did that in class, achiral. Mirror, piece of rice has a superimposable mirror image. That's achiral. Now, baseball glove, for those who haven't played baseball, there's one made for your right hand, just like your winter gloves, and one for your left hand. So baseball glove is chiral. Now, we did in class your face where I said, touch your right cheek. Well, how do you know which is right or left unless it's chiral? It is. And a car, and if you remember, I did my great artwork. A car is also chiral. It has a non-superimposable mirror image. Now, I don't have on there, but let's try two more. How about a pair of pants with a zipper in front and pockets? Is that chiral or achiral? I'll give you a few seconds to think about it. That's my few seconds waiting. All right, think about it. A pair of pants with a zipper in front is chiral. How do you know? Well, do you have a right and left pant leg where it made a difference? And if we were in class and I got sweatpants on, since I've gone to Zoom, I rarely wear Dockers or anything like that. I'm comfortable at home, so you can see anyways. But anyways, think about it. You put your right leg in the right pant, your left leg in the left pant. Now, if you put them on backwards, and I told my class when we're face to face, you see me wearing my pants backwards, please get help for me. I'll need it quick. But anyways, it won't feel right. The zipper in the back, when it's supposed to be in the front, is chiral. And if you do a mirror image, it's non-superimposable because the zipper and everything, and the way the pants pockets and everything, and the way it's cut, it doesn't go on right when it's backward. Same thing if you put your right shoe on your left foot. It's not going to feel good because your feet are chiral and your shoes are chiral. All right, let's continue on. Next thing you should know, circle the chiral carbons in the following molecules. How do you know something's chiral? It has 4 different atoms, 4 different groups, or a combination of 4 different atoms and groups. If it has 2 or more of the same atom or group, or if it has, if it's a carbon in a double or triple bond, it cannot be chiral. So if we look at A, the first carbon has 3 hydrogens, that's out. This carbon with CH2 is out, the CH3 is out. Now, if we look at this carbon with a chlorine, hydrogen, methyl group, and these 3 carbons with the chlorine hydroxyl, 4 different groups, it's circled. If we look at the carbon with the chlorine and hydroxyl group, that plus the methyl and these 3 carbons with a chlorine, 4 different groups. Now, if we look at B, what do we have? Well, immediately the CH3 is out, 3 hydrogens, CH2, and the 2 carbons in a double bond. That leaves the carbon with the thiol, SH, methyl. Oh, by the way, CH2 double bond is called allylic group, and you have 4 different groups. Now, if we look at C, we have carbonyl, carbon double bond oxygen out. Each one of these carbons, these 3, has CH2, that's out. This is CH2, CH3. Remember, 2 or more of the same group atoms, it can't be. But this carbon has a hydrogen, ethyl, these 2 carbons from the ring, and these 2 carbons, which are different. And same thing here. And let me give you, let's see, and I did give you one here. Let's look at E. Remember, CHO is an aldehyde. And therefore, that carbon can't be chiral because it's in a double bond to oxygen. Uh-oh, my voice is thick. One time with general chemistry, I lost my voice and I was doing a review. I did the whole class just writing on a white, actually it was a chalkboard, it was so long ago. Not a fun night, but hopefully I won't lose my voice today. And if we look at the bottom carbon, CH2, 2 hydrogens, and each one of these intersections in a Fisher projection is a carbon, and they each have 4 different groups or atoms. And let me give you one to try on your own, because I want to share today. And everybody see on the whiteboard the molecule I just drew? Circle the chiral carbons in that molecule. 3 points each. And when you're done, give me a thumbs up. Excuse me again. And for those who finish early, please be patient. I try and give everybody time to finish up. All right, let's do this. And the question is, which are the chiral carbons? Circle them. Well, as soon as you see 2 hydrogens, that's out. Aldehyde, remember when you see RCHO, that's the same as writing it this way. My software and sugar chemists write it this way, so that's the way you might see it on a test. So that's out, because it's in a double bond. That leaves this carbon, and notice hydrogen, hydroxyl, aldehyde, and the rest of this, 4 different groups. Oh, let's look at this carbon. It's got a hydroxyl group and a hydroxyl group. It's out because it has 2 or more of the same group. It can't be chiral. Now, let's look at this carbon. I'd circle it because it has hydrogen, hydroxyl, these 3 carbons, and these 2 carbons with a bromate. It's different. It's chiral. 4 different groups or atoms or combination thereof. And finally, this one, bromine, hydrogen, the CH2OH, and all the rest of this. So if I clean this up a little, those would be the three you would circle. Now, one thing I have mentioned, it won't be on test, but I thought I'd tell you now. This is glucose, which is the sugar in our blood, and what I'll talk later about it. And it has 1, 2, 3, 4 chiral carbons. Now, this will never be on test, but the number of isomers for a structure equal 2 to the n, where n equals the number of chiral carbons. So for isomers of glucose, n is 4, which is 16. There's 16 possible isomers, another enantiomer, and other types that are just different stereochemistry. Mother Nature only makes and picks one, which is always an amazing concept or a fact to my mind that there's 16 possible and only one she picked the right one that works for all of us, which is interesting if you're an organic chemist, which I am. All right, let's move on. Now, next one, draw the enantiomer of the following molecules. What's an enantiomer? A mirror image of the molecule. And therefore, what I'm really asking you is draw the mirror image of the following chiral molecule. Now, I don't play games on tests. I know some instructors will write down, and the answer is, well, it's not chiral. It doesn't have a non-superimposable mirror image. Therefore, you can't write the enantiomer. No, if I ask you to draw the enantiomer, there's an enantiomer. So how do we do it? Well, I like to put in my imaginary mirror what's close to the mirrors you see first. For the achiral carbons, the aldehyde CH2OH, you don't have to put it if you want to. And here's the mirror image. And let me have you, and this one, oh, B cross off. That will never be on a test. Sorry, I used to teach that and I forgot I don't do that anymore. All right, let me give you one to do. Oh, spell enantiomer wrong. I don't know if this exists in nature, but why don't you draw the enantiomer of that compound? And when you're done, you know what to do. Thumbs up, please. All right, I think everybody's done. So what's the enantiomer? It's the mirror image, because I'm always going to give you a chiral compound to do this with. So you don't have to, but I like to put my imaginary mirror in there. It helps me. Here's the backbone. Those carbons at the bottom, this is CH2OH. If you wanted to draw it this way, that's okay too, but you don't have to because this is a chiral. Same thing here. If you wanted to draw it this way, I'd smile and say, you didn't have to do that, but it's okay. But now these carbons are what's closest to the mirror, H. What's farthest away, hydroxyl group. What's closest to the mirror, bromine. What's farthest away, hydrogen. What's closest to the mirror, hydroxyl group. What's farthest away, hydrogen. And this would be the enantiomer and your answer. Now, here, give the organic product or products. When I say give the structure of glucose and fructose, I stopped doing that, but you don't need to know fructose and glucose unless you want to. Now, here you can either give the product, draw it, or write down the name. And A is if you take fructose and glucose and react together with acid, what do you get? Well, fructose and glucose bond together, gives you table sugar known as sucrose. Now, next, if you take a lot of glucose and react it with more glucose, the presence of acid, and Mother Nature uses an enzyme, which is really just most of them are acid catalysts, very fancy ones, you get starch. And as I did the other day, I showed you how the cyclic form of glucose links together in acetals to form starch. And the key reaction for that, and when I did this, I wasn't good with the software. I've got this more rounded off, that line. If you take the cyclic form of glucose or any hemiacetal, react it with an alcohol and acid, here's the general reaction. You replace the hydrox group with OR triple prime. In this case, OR triple prime is ethyl, and that's the molecule you get. And when I handwrite it, the bond. By the way, this line from oxygen to carbon is a carbon-oxygen single bond, but since I don't tell anybody it was nice, I gave you a gift, you didn't have to learn Hawthorne projections. This is my way of making it easier for you. And here I have a different R group, cyclohexane ring. Now, what happens when you react sucrose with acid and water? Well, sucrose is held together by an acetal linkage. You get back the aldehyde or ketone plus the alcohol you would have used to make that. Well, in this case, sucrose is glucose and fructose held together. And when you add acid and water, like in your stomach, you get glucose and fructose back. Now, starch is a polymer of acetal linkage of glucose. And in your stomach, when you digest it, acid and water, acid is hydrochloric acid, breaks it down to glucose molecules. Now, G, just forget about this. This is a specific type of starch. Okay, first of all, any questions on the problem set? Looking at the chat, listening to my speakers. All right, let's go through some important stuff that'll help you for test 4. All right, leaving my class, you should know how to describe with words and a general reaction what happens when you eat something that has a fat in it or fat molecules or oil. And what's the functional group you should know in fat and oil? And that's ester. It's a triglyceride triester. Therefore, how do you describe with words and a general reaction what happens when you eat a fat or oil? Well, it's acid, hydrolysis of an ester. And here, excuse me, a general reaction you learned for test 3, ester, acid, water, you get the carboxylic acid you would have used to make that ester, plus the alcohol you would have used to make that ester. I just thought of something. I was never been curious how our body uses fatty acids or glycerin to do things. Well, if I have time this weekend, I'll look that up or explore that. All right, now you should know this. Next one. By the way, this means end of line, the way I do it, something new. How do you describe with words and a general reaction when you eat a carbohydrate? And here I'm really talking about starches as the carbohydrates. And what's the functional group that holds glucose together in carbohydrates? And that is an acetal. Therefore, when you chew it and swallow it and goes in your stomach, you have acid and water, acid hydrolysis of an acetal. And the general reaction, which I'll let you write for both an acetal and ketal, you learn for test number 2, acetal or ketal, R prime can be R prime or H, acid and water. You get back the aldehyde or ketone you would have used to make that acetal, plus you get back the alcohol. You don't have to put the 2 there, but I will, just because some people get upset if I don't. I don't need upset students. Now, in real life, both of these are in the same molecule connect together, we call glucose. Now, important thing you should know leaving my class is you should know how soap works. And the first thing is you should know what's the structure in general of soaps. And it has a nonpolar tail and a polar head. And the most important thing, how soap works, and in a whiteboard, I do the same thing. If I can spell it right. Let's try that again. Like dissolves like. That's the key to the whole puzzle, how soap works. Now, like dissolve like, we talk about polar things are soluble in polar things, non-polar things are soluble in non-polar things. You mix 2 together, they are not soluble. And what's polar? Water. What's non-polar? dirt, and grease. A lot of other things too, like vegetable oil, gasoline, Vaseline, baby oil. But for this class, polar, water, nonpolar, dirt, and grease. If you had on your hand, greatly magnified, a piece of dirt, water looks at dirt and says, I'm polar, you're nonpolar. I know light dissolves like, I want no part of you. But if you now wash your hands with soap and water, the soap molecules, the nonpolar tail, are attracted to the dirt because light dissolves light. And I'm only going to show a few soap molecules, but it totally surrounds the dirt particle. And this new combination of soap and dirt, the way I have it drawn, is called, and you should know, this is a micelle. And once we have a micelle, on the outside, totally surrounding the dirt, are the polar heads. So now water looks at this and only sees polar heads and says, oh, micelle, I'm polar, you're polar, let's go down the drain together. And lo and behold, my hands are clean. And when I took my shower this morning, I was probably the only one in the whole area around my house was thinking, wow, I'm making micelles now to clean me. And I was. And you should know this. You should know what is the structure of soap, non-polar tail, polar head. You should also know the key to the whole puzzle is light dissolves light. And you should know that water is polar, dirt and grease are nonpolar. When you have soap and water, you form a micelle, which looks like this. And a micelle, because the polar heads are on the outside, now look polar to water. And water says, oh, I'm polar. I bet you didn't know water can talk. It can't, but it's nice to think that way. I'm polar, you're polar. Let's go down the drain together. And the same thing happens when you use your washing machine or if you have a dishwasher. The soap and water form micelles, and that's how it gets things clean. All right, another thing you should know. Test number two, aldehyde or ketone plus an alcohol plus acid. This attacks that, and you form an acetal or a ketal. What's attached to carbonyl carbon is now attached to this carbon with the hydroxyl group. You need acid catalyst when it's between two molecules intermolecular reaction. When it's all in the same molecule, you don't, and you have usually an equilibrium. And this is C5, 1, 2, 3, 4, 5. This is this alcohol, and here's our carbonyl. And this will attack that and bond to it, just like right here attacks it. And therefore, what's unaffected by the reaction still is there because you don't break carbon-carbon single bonds or other atoms in this case. Now, this oxygen, here's this carbon, is this carbon right there. This red bond is this right here. And this carbon will have an OH group. And you should know how to do this. If I give you this structure, you should be able to draw this. If I give you this structure, you should be able to draw the starting material. Because this is the first key Mother Nature uses to form all the starches and cellulose in our life. This is for glucose. Let's look at fructose. And I show the carbonyl, this carbon right here, but it just might be a double line to oxygen from the line itself. But this is fructose, the sweetest sugar around. And same reaction. When it's intermolecular, you need an acid catalyst. Again, this attacks that. And you form an acetyl ketal. Now, this one is intramolecular in the same molecule and it's still C5. Remember, 1, 2, 3, 4, and this is 5. That's how you count to give the carbonyl the lowest number. And here again, same type of reaction where this attacks this. Here, this hydroxyl group is this hydroxyl group on the carbon alcohol, and it attacks that carbon. And when it does that, what's not involved in the chemical reaction, if I can draw a straight line, let's try this again. That's better. I'll put this carbon right there because I know I'm going to do anything, but this and this is not changed. This will not. I have this oxygen, which is this oxygen, and it's bonded to the carbon with the hydroxyl group. And there we go. And if that didn't happen, we wouldn't have table sugar. That would be sad. We wouldn't be able to sweeten things up when we cook. And here again, you should know, if I give you this structure, you should be able to get to this structure. If I give you this one, you should be able to either write fructose or draw the structure of the starting material. And those are important for test 4 and the final even. All right. Now, yesterday I was going through stuff in carbohydrates, and I believe we went through, let me take a look at my, if I have it open. think we went through a lot of it, but let's just do a couple more things just to remind you. Everybody see sugar on your screen? Is it sweet? Oh, bad joke. But anyways, you should know. Oh, I don't have it here. I have it later on. That sucrose, you don't have, here's the structure, but you don't have to know that. Sucrose is glucose and fructose bonded together. And when you have a lot of glucoses bond together, you make starch. And I showed you that in the problem set. Well, let's review that again. If I take glucose and fructose, what do I get? Sucrose, table sugar. If I take a lot of starch, or glucose, I mean, plus some more glucose, and react it with acid, I will get starch. And Monday's lab will deal with how do you know if there's starch in something? Not carbohydrates, starches. There's a difference. Now, if you have sucrose and you eat some acid and water, what's the product? Either draw it or write the name of the product or products. And this one, I'll help you out. The two products you get when you react sucrose with acid and water. And the answer is, guess what? You get glucose and fructose because your stomach breaks it back down to the original monosaccharides. Now let's do one more. Either draw the structure, write the name of the compound that you get when you react starch with acid and water. That means if you had a piece of bread for lunch today, what's going on in your stomach right now? Or if you had stuff that Dr. White doesn't eat. In fact, last weekend I was over at my best friend's house. He just moved back from Indiana to Illinois, and we had hot dogs with French fries. I haven't had French fries in many, many years because I don't need them like a hole in the head. I love French fries. Oh, they were good. All right, what do you get? You get glucose. And that's what you get. If I look at the time, guess what? It's time for a break. Take 5 minutes, come back at 1:55, and we'll continue on. Plus, I'll introduce you some new stuff. And then today, I'll give you something most people have never seen, and that's the inside of a chemical plant. Now, I'm going to ask one of you a personal question, if you don't mind. Matt, have you ever been in a chemical plant? Okay.
Yeah, with my job, and my dad did some stuff with that too.
Okay, I wasn't sure if you did or not, but I bet you never have been in one with, well, you'll see. And I'm going to take a 5 minute break. I'll see you in 5. Time to get going again. And before we go on to the new chapter, amino acids, I thought I'd take you to somewhere, talk about, if I can find it, there it is, it's been hiding. All right, everybody see on the screen cellophane? Thank you. Now, what is cellophane? You know that crinkly stuff you get like candy in, or sometimes crackers are in that clear plastic-like material that's real crinkly? Well, how do they make it? This is chemistry I've never been involved in. This is one of the few things I can say. Right here, this is cellulose, which is a polymer of glucose that we can't break down. The oxygen bonds in cellulose are top and bottom. It's called alpha beta. In glucose, in starches, it's not alpha beta. It's all, I think, beta. But cellulose is from plant material. You can buy it cheap. And if you react it with sodium hydroxide and carbon disulfate, you replace some of the hydroxyl groups with this, I don't even know the name of that functional group, but it's akin to like a carboxylate anion. And this is that material. And that's how they make it. One of the things in chemistry, when you're in industrial chemistry, if you can find a really inexpensive starting material, that's something you want to work with. And cellulose is very, very inexpensive. Now, years ago, back in the 1900s, they found a certain reaction and that's nitrocellulose. And what you do is you take cellulose and react it with nitric and sulfuric acid. A reaction I didn't show you is you learned it for a benzene ring, but never ever try this at home. Take, say, sulfuric acid, H plus nitric, and you nitrate it. And I don't think there's an oxygen there, but let me take a look back. Again, this is chemistry I've never done other than the lab. Yep, nope, I was right with the oxygen. And this is making nitrocellulose. Now, this has been, when they first made it, depending on how many nitro groups you put on it, made a nice, flexible, clear plastic. And guess what they did? They put photographic chemicals and they made the original film. You know how when you go to the movies, the old days, I don't know now, I think it's all digital, but I might be wrong, but they played a film through the projector. Well, those are made of nitrocellulose. Unfortunately, nitro groups like in trinitrobenzene makes this compound with age unstable. And by the 1950s or 60s, movies from the old days, the 1905, 1910, were catching fire, blowing up. And I think a number of producers who are well off started a group that went and took those old movies on nitrocellulose and recopied them onto more stable media, and that saved them. Now, one other thing I like to talk about is cotton. And everybody see cotton on their screen right now, right here? No. All right. It's a polymer of glucose. But you can't eat it. It won't. Now, if you notice, cotton here, where my mouse cursor is, has a lot of hydroxyl groups. And let's talk about how many of you have ever heard cotton's a nice fiber because it breathes? Well, guess what? It's not taking in oxygen. What they mean is this can hydrogen bond to water, and such as your perspiration. And that wicks moisture off your body, which makes life more comfortable. And that's how what we mean by cotton breeze. Now, one of the things I do want to teach you is, have you ever wondered, oh, I've got one here, it's a microfiber, how your hands are wet and you go like this and they're now dry. And you can do the same thing with a paper towel. How does that work? Well, the cotton, and I'll just do it like this, has hydroxyl groups, which has a slight negative, slight positive charge. Water has the same dipole moment, it's called, and it forms a hydrogen bond. If I could spell it. Today's you can't spell anything day. I've been pretty good this summer. I haven't lost my. And that's how water gets magically pulled off your hands. When you're wiping your hands on a towel or a paper towel, which is paper is the same thing, cellulose with hydroxyl groups, you're forming hydrogen bonding. And that will help you get the water off. Now, how many of you have ever had towels with time, they go bad on you? What I mean by bad is they don't get the water off as well as they used to when they were brand new. And the problem is if you use a fabric softener, the fabric softener will be attracted to the hydroxyl group, and the long alkyl chains make that look nonpolar. And water doesn't want anything with nonpolar. So how do you get your towels back? By the way, that property is called re-wet. I've worked in those areas with fabric softeners, so I know about it. And how do you get it back? Well, simple. You wash your towels without using a fabric softener, either in the washer or the dryer. Do it once or twice, and they'll absorb just like they were brand new. Now, one other thing I wanted to talk about, long time ago, it seems that way, I taught you this reaction. Take an alcohol, react with ethylene oxide, and you get this ether alcohol. Well, it turns out that reaction is a very useful one to make a product a lot of you have in things you buy. And what is that product? As hydroxyethyl cellulose. I think I mentioned it once, twice, or 100 times. Wikipedia has been so heavily policed by our chemists, especially organic chemists, you don't find errors. Well, on here, you should be saying hydroxyethyl cellulose. Here's the structure. And what they do is they take cellulose, plant wall material, and react some of the hydroxyl groups with ethylene oxide. And you get, or R is CH2, CH2OH. And N is on the order of about 1,000,000. So you have these groups. Now, what is hydroxyethyl cellulose good for? It's a very good thickener. And it will take water and make a gel out of it. And where do you want to do that? Well, certain products you want thick. And I remember I said I would get into things that you'd never think I'd get into. And here's one I talked about briefly a while back. Well, no, I haven't. All right, let me see. Does everybody see on your screen KY jelly? And it's a lubricant for a personal thing, activity lubricant. I'll let you figure it out. And what it really is glycerin, which is not that thick, and water and hydroxyethyl cellulose and other things that are preservatives. And what makes it thick is water with hydroxycellulose. It thickens it up real good. And I've worked at products where you use hydroxyethyl cellulose to thicken up things. And you got to be careful how you mix it and everything. Otherwise, you get these goops not truly spread throughout. And that's some interesting things I thought I'd teach you about what you can do in chemistry with starches or really carbohydrates. Next time you wipe your hands on a towel or paper towel, think about it. You're doing hydrogen bonding of water to that paper towel or towel to get the water off your hands. All right. Any questions about anything that might be on test number 4 before I move on? All right, let's continue on. All right, does everybody see chapter 17 on the screen? This will not be on test number 4, but there will be some points on the final exam covering this. Now, this chapter deals with the last food group we haven't talked about yet. And it deals with amino acids, peptides, and protein. Now, what is an amino acid? Well, it's an organic compound that contains an amine, which we call an amino group, and a carboxylic acid. And instead of calling it an amino carboxylic acid, the name was shortened to amino acids. And here's the structure of all amino acids. A better way to draw it would be this. Now here we have a carbonyl carbon, and when I taught you what ketones, and it's also true with other things like esters and carboxylic acids, the carbon attached to the carbonyl carbon is called the alpha carbon. And in all amino acids, an NH2 group, an amino group, is always on the alpha carbon. Notice you have a carboxylic acid group and an amine group. And you put them together and you get a amino acid. And really it should be called an amino carboxylic acid, but organic chemists are lazy. We are. Now, switches on. First of all, you should know how to draw an amino acid in general. Now, chirality. Remember you learned chiral, carbon, 4 different groups to carbon, 4 atoms or 4 groups or combination thereof. Now, the alpha carbon of amino acid is bonded to 4 different groups. The alpha carbon, this carbon right here, has a hydrogen amino group, a carboxylic acid, and R. When R is not hydrogen, then it would be CH2. The alpha carbon is chiral. When R is not hydrogen, the amino acid is chiral. And most amino acids are chiral. By the way, and I'm not going to ask you to learn any amino acid names, but when R is H, that's called glycine, and that's, I believe, the only amino acid that's not chiral. Of all the things, functional groups and things I've worked in chemistry, I've never worked with amino acids other than that's what my body breaks down certain things to make. Therefore, amino acids exist in 2 enantiomeric forms, mirror images right and left, which means our DNA is chiral. RNA too. Now I'm way beyond my level of expertise. Now, couple things, switches off right now. Acid-base properties of amino acids. I can go click, it's off. Amino acids contain a basic amino group and acidic carboxylic acid group. And depending on the pH, when it's around 7, we have what's called a Zwitterion. By the way, Americans call it Zwitterion, but because I speak German and the word Zwitterion comes from German, I pronounce a W like a V, which is how you do it in German. And Zwitterion comes from the base Vi, which is German for 2, ZWIE, or is it EI? I think it's IE. So Zwitterion means 2 ions. And what is that? Well, what happens is at pH about 7, this is a base, this is an acid, and you get this equilibrium, and this is your Zwitterion right here. Switches off. And it's something quite ingenious Mother Nature does, because now you've got a polar compound, which makes it more water soluble, like in your stomach and blood. Now, I'll again switches off, but in case you learn this, need to know this for other classes, a Zwitterion is a molecule that has both a positive charge on one atom and a negative charge on another in the same molecule, but no net charge. If you look back here, here's a plus, here's a -1, add it together, no net charge, 0. Now, switches on full time. Full time, that makes no sense. Switches on full blast, that's better. And let's look at this reaction. But before we do, for test 3, you learn the following. A carboxylic acid plus an amine will give you an amide. And remember, our prime and our double prime can be hydrogen. Well, with that in mind, notice, Dr. White got really fancy. Here I have my carboxylic acid. Here I have my amine. The R prime is this whole thing on the amino acid, the second one, and one of the R double prime, you call one of the hydrogens. And carboxylic acid, amine, and notice we have an amide. This is an amide functional group that's holding 2 amino acids together. And if you look at the end here, I have more amino groups that I can react more amino acids with. I have at this end more carboxylic acids, which I can react more amino acids with. And think of it as a polymer growing, or like a freight train, you're adding boxcars to it. So the key thing is, when you react 2 amino acids together, you form an amide. Switches off because I'm not a protein chemist. For some reason, protein chemists call that bond, which I call an amide, a peptide bond. And I'll never ask this on the final or any test, but the covalent bond between the carboxylic acid of one amino acid and the other of the amino group of another is called a peptide bond. Dr. White still calls it an amide bond because I'm a synthetic organic chemist, not a protein chemist. Now, a peptide, and again, switches off, but I thought I'd teach you this, is a molecule containing 2 or more amino acids, which the amino acids are joined together in an amide bond, which everybody else calls a peptide bond. Now, what's a protein? Well, protein is a peptide in which at least 50 amino acids are present, connect together by, they call it peptide bonds. I call it amide bonds. So proteins are just amino acids, and so on, and amino acids. And essentially, what is a protein? It's a polymer of amino acids. I guess you call it a condensation polymer. Never thought of that. Wow, that's something I just came up with. Now, this switches on, click. What happens when you digest something with protein? Like I had for lunch today. When you consume protein, your stomach and intestines break it down to amino acids by acid hydrolysis of an amide. Here we have, you learned for test number 3, if I have an amide, and remember R prime and R double prime can be hydrogen. In your stomach, there's hydrochloric acid and water. And now when I react that, what do I get? I get back the carboxylic acid I would have used to make that amide, plus I get not the amine, because amines are basic. In the acidic environment, you form the amine salt. And that's how your body breaks it down. It just so happens the amine group and the carboxylic acid and amino acid are in the same molecule. And what does that look like for a peptide? Or if we're longer, a protein, here's our amide. And what do you have? Switches on. You get back the carboxylic acid you would have used to make that. That's this. And then you get the amine salt. You would have used to make, of the amine you would have used to make that. Remember, R prime and R double prime can be hydrogen. And here you get the amine salt right here. But let's look at this one right here. This is on at the very end, you're going to get an amine, but in the presence of acid, you form the amine salt. Then we went through that for test number 3. And this is how your body breaks down. If this were longer, a protein, you get the amino acids, and that's how your body breaks it down. Then I have no idea how it puts it back together the way to make things like your skin and other things. All right, switches on full blast. Leaving my class, I've already talked about today. You should know how to describe with words and a chemical reaction what happens when you eat. I'll do it again. I'm not going to cheat you out of it. Eat of fat or oil or some food with that. And what's the key functional group here? Ester. And how do you describe that? Acid, hydrolysis of an ester. And what's the general reaction? You learn for test 3, ester, acid, water. You get back the carboxylic acid plus the alcohol you would have used to make that ester. You should also know what happens when you eat a carbohydrate. And what's the functional group that holds glucose together in carbohydrates, meaning starches? And that's an acetal. And in your stomach, there's acid. So it's acid and water, hydrolysis. Of an acetal. And what's the general reaction? You learned this for test number 2, acetal or ketal in general, acid and water. The carbon with the two oxygens becomes a carbonyl. What's attached to it stays attached to it. Plus you get back the alcohol you would have used to make that acetal along with the aldehyde or ketone. And for those who must, I'm trying to make it even smaller so it's almost invisible, but anyways, you get that. And now the last major food group you should know. What happens, how do you describe with words and a general reaction when you eat something with a protein? And what functional group holds amino acids together in a protein? It's an amide. And when you eat it and you swallow it and goes in your stomach, there's acid and water, so it's acid hydrolysis. And what's the functional group? An amide. And how do you draw the general reaction? And you learned this for test number 3. Remember, you don't have to write this down, but I will. Amide, acid, and we'll use HCl, and you'll understand why in a second, because I need to count the anion. You get back the carboxylic acid you would have used to make that amide, and you get back the amine salt of the amine you would have used to make that amide. Now, in amino acids, those are hooked together. And now we've covered the 3 major food groups. And let's go through it again. When you eat a fat or oil, what functional group is in there? It's an ester. And when you eat it and consume it in your stomach, and I think your small intestine, but don't quote me on that, you have acid. How do you describe it? Words and general reaction. You have acid hydrolysis of an ester, and here's your general reaction. Ester plus acid and water, you get back the carboxylic acid plus the alcohol you would have used to make that ester. When you eat a carbohydrate, what is the major functional group that holds glucose together in a polymer for starches? And that is an acetal. And when you eat it and swallow it, in your stomach is acid and water, and that undergoes acid hydrolysis of an acetal. And here we have an acetal or ketal, acid and water, and you get back the carboxyl wrong, I'm ahead of myself. You get back the aldehyde or ketone plus the alcohol you would have used to make that acetal or ketal. And finally, when you eat, and we learned today, a protein, what's the functional group that holds amino acids together in a protein? And that's an amide. And when you eat that, you have acid hydrolysis of an amide. And what's the general reaction you should know? That's an amide plus HCl. That's what's in your stomach. I think it's always one of the miracles of life that I and you too have cells in your body, your stomach, that synthesize, make hydrochloric acid. Amazing. But anyways, you get back the carboxylic acid and the amine salt of the amine you should have used to make that amide. And those are, leaving my class, you should know those 3 things. And you should also know how soap works. That's important. Now, if I look at the clock, Let's take a 5-minute break a little early. You'll understand why. Come back at 2:36. I'm going to get up and stretch, and then I'll have a very nice surprise for you. Let's get going. All right. First of all, I've covered everything new I'm going to cover this semester. Remember the first day of class, I said, this semester, you'll find out my world of organic chemistry is your world of organic chemistry. And now you have. And you have knowledge that most people in the United States or on our planet do not have. And I think from what I've seen, test scores, you've got a pretty good knowledge of it. Now, next week, excuse me, I will be doing a review for the whole semester. I will try and get it by maybe tomorrow, set up the final exam points breakdown information. For sure by over the weekend, I'll have it up. And then I'll talk about that on Monday. Monday, I will go through the test number 4 answers because I'll have that done. But I will cut it out of the video. But the rest of the week, next week, which will be Monday, Tuesday, and Wednesday, I will do a complete review of the whole semester. It helps students. They tell me that, and I can see that on the final exam. Now, one thing I will do on Monday, which I do, is once we have all 4 tests, you have your scores, I'll show you what you need to get on a way of calculating what you need to get on the final to get the grade you want, A, B, C, or so. Remember, Monday, the extra credit project, it's 10 points. Do it. It will help your grade. No matter how good you're doing, it never hurts to have 10 points in the bank. So I'd highly recommend you do that. Remember, if you're not sure, or if you want to look, have me check it before you upload it. I will check it and tell you this is wrong, you got to change it. And there's no reason why you shouldn't be able to get 10 points. Also, all of you are hearing my voice right now. If you're missing any labs, don't forget about my amnesty program. If you don't have it, email me email me what labs you need, and I'll send you those so you can email them back because the link in D2L I've closed, time closure. All right. Any questions before I go into some fun stuff? Look at the chat. Nope. All right. I've been very lucky to have a very long and distinguished career in the chemical industry. If you have any questions, see me tonight. I hope you enjoyed the tour. People don't get to see those things. It's an exciting place, dangerous, but exciting. And with that, I'm done. Don't forget, tomorrow we'll have our lab. And then right after the lab, about, I don't know, by 2 o'clock, I'll let you go because we had our long session today. And then I'll send out the password for test number 4. And with that, gehen gesund. Janet, my office hours are from 5 to 6. If you need me to stay a little later, I will. Can you make that? Okay. And with that, I'll say, gehen gesund. Goodbye now.